This is another little short video on gene therapy, a follow up to the previous video, part two. Here we have a diagram that is showing, it says it's a genetically engineered virus. This is our viral vector. And if you look close at the diagram here, it is showing that a normal hemoglobin gene is being inserted into the genetically engineered virus, which we call a viral vector. That viral vector is then injected into a human being, and it is delivering the corrective gene to the bone marrow by way of the viral vector. If you look here, it says a normal hemoglobin gene is inserted into a modified virus, which is a viral vector, and it's used to insert the gene into a cell with an unhealthy version of the gene. Now, in class, we read this passage about gene therapy, and basically, again, gene therapy is designed to introduce genetic material into cells to cure a genetic disease, to make a correction to a mutation. Many of these mutations are point mutations, they're very small. These aren't major chromosomal mutations. And here again in the passage, if you have this in your notes, it says that certain viruses are often used as vectors because they can deliver the new gene by infecting the cell. Okay. And here we have a diagram using stem cells as well as a viral vector to deliver the correction for the gene. Now, this diagram is showing us a picture of how this occurs in somebody who has sickle cell disease or sickle cell anemia. And as we've discussed in previous units, sickle cell anemia is a genetic disorder. It is passed down from parent to child. It is a mutation that changes the shape of the blood cells into little sickle shapes, and it causes many problems in the body. So instead of delivering therapeutic medications, instead of treating the disease in other ways, this type of therapy treats the disease at the cellular level, at the level of DNA. Now, how is this different from creating GMOs? You're not inserting in gene therapy. You're not inserting genetic information from another species or another organism. You're only inserting a correction. And so when we have people who receive gene therapy, they're only receiving the correction for the gene. They're not getting DNA from another species, therefore creating a genetically modified organism. It's just in a more improved way of delivering a correction to somebody who has a genetic disorder. They're still experimenting with this. It's still a new study, and it's been successful in many cases. So again, gene therapy is introducing genetic material that is from the same species, human genetic material, into cells of abnormal genes that are coding for a mutation that is causing a disease, cystic fibrosis. In this case, this would be sickle cell anemia. Again, they are genetic disorders. They don't cure viruses or they don't protect from viruses. That's completely different from that. So, if you have any questions, you can review the written material. And again, look at these diagrams. I always say annotate. That is going to help you discover and find differences if you can look at a document and draw the information from there.